Hello and welcome back to part 2 of week 1's lecture for Organismal Physiology, Biology 320 at Portland State University. And we're moving on from basics and study of physiology to talk about homeostasis and beyond. So with that, let's begin. So one sort of overarching theme of homeostasis is that with regard to the parts an organism has, how it's built, organisms are structurally dynamic, meaning that they can change in the short term, including changes like changes to what's in the cell membrane, but also building blocks of an organism. So for example, cell turnover. All the cells at the surface of my skin right now are not the same ones I had 2 months ago, and yet my skin is my skin, right? So although organisms change over time, the organization is stable across time. That's why although I change which cells I've got, I stay me and don't turn into someone else or a pile of goo. Most of the cells of a multicellular organism are exposed to the internal environment, not the external. Which means another way to say this is organisms have a defined, defended, and stable edge. And your edge is important. So you might be wondering, like, why is she speaking so reductively? Why is having an edge and a middle important? Those are kindergarten words. Your edges are what keep you distinct from the environment you exist in, and what helps to keep valuable things inside of you and keep harmful things out. So valuable things like water and nutrients, harmful things like pathogens and extremes of temperature. So although I am being reductive in my language, having an edge is critical. So homeostasis is centered around the idea that organisms have some choices about the relationship between their inside and their outside. So because most of your cells are exposed to the internal environment, not the external, the way that the interface between external and internal works is going to mean that you either try to match the environment you're in or you don't. So take me, for example. I was warm earlier because I was on the exercise bike inside. If you're listening to this in the future, this is being recorded during the Oregon wildfires, during COVID, during quarantine. So I'm in my basement on my exercise bike, watching true crime television, sweating my skin off because I've raised my internal temperature, but my cells don't work well if my temperature raises above 37 degrees Celsius. So my sympathetic nervous system turns on my sweat glands to push water out of me and induce evaporative cooling. So I'm trying to keep my internal environment constant at 37 degrees Celsius, even though I'm generating a bunch of heat and heating up. That's one example. Speaking of temperature, let's look at some charts. So you might be looking at this right off the bat and thinking, conformity, well, the flat line looks like conformity because it's flat, but you would be wrong. So if you look carefully, you'll see external and internal temperature plotted here against each other. So external and internal temperature. If an animal is a ultimate conformist from a thermoregulatory perspective, then whatever's going on outside, the animal's going to match on the inside. So if it's 20 degrees C inside, 20 degrees outside, 30, 30, so on and so forth. So we have this direct linear relationship between the two. If the animal is a regulator, then regardless of the external temperature change, and if you understand centigrade, you'll note that 10 and 40 are very different temperatures, and yet this animal is holding itself steady at, oh look, surprise, 37 degrees C, which is human body temperature on average. Now, I will say, it's not always a perfect 37 degrees centigrade. It really kind of does this. But if you were to do a linear transform of the average of those values over time, you would get a straight line. So that's another important thing to keep in mind. The graphs like these are reductive for clarity. They don't represent the absolute truth, rather a representation of it. Also, interpreting graphs is something that you should be able to do. So if you're rusty at it, practice makes perfect. And a good way you can achieve that is by reading primary literature and trying to understand the graphs, which are often very complicated. Okay, so here we have another example of temperature conformity. So there's a linear relationship between blood temperature and water temperature, making this critter an ectotherm. But we've got a different situation when it comes to chloride regulation. So chloride is an important ion. It's exchanged with sodium and it's exchanged with bicarbonate in a lot of vertebrates to keep cell pH and cell tonicity stable over time. And so because it's an important anion, you don't want it to vary widely within an organism. So even though water concentration of Cl is increasing, so down here it's something like 0, and this would be like milliosmoles or something like that. There's lots of ways to express concentration. You can see that this is increasing, but the salmonid is going to keep its chloride relatively steady. Now again, it's probably going to do something like this, because the fish is going to be ejecting chloride as it gets closer to this end of the graph. It also has kidneys, and it's going to pee out chloride. So both the renal system and the gills are going to be excreting chloride such that it remains stable even though the water concentration is increasing. And that's important because, of course, think about making preserved meats or pickles. What do? Well, you stick whatever you're trying to preserve in either just a bunch of salt or really salty water, which preserves it by preventing organismal growth and success, but you can't really be alive in those circumstances. So, why not always be a regulator? Wouldn't it be easy to just do whatever your environment is doing? Well, or excuse me, wouldn't it be easier to just regulate every single thing? The answer is no. Sometimes, I got my argument backwards, I apologize, I've been talking for a long time. That would mean that you would have to regulate every single little thing. And regulation, it costs calories. So the way that the fish is doing this, by the pumping of chloride, costs ATP. So regulation is energetically expensive. Another reason is that environmental conditions on Earth often are very extreme. Extreme to the point where you can't really regulate against them. So if you can instead adapt a way to survive in spite of change, that's good too. And most organisms really do both. They're going to conform in some ways and regulate in others. Also, the reason why not be a conformer, you could ask the same question. If you were a 100% conformer, you would be functionally no different than your environment. So that's also not ideal. You need a mixture of the two. So, homeostasis. We're circling back to that word. Essentially, if you're an organism and you want to stay alive and stay the creature that you are, more or less, you need to keep your internal state relatively constant compared with the external. And this requires physiological processes, so chemical reactions, pumping, movement of fluids, and that helps you retain internal constancy even if your external environment is fluctuating wildly. So for example, think about the very dry desert and the profound difference between daytime temperature and nighttime temperature. One example, there are many. So for regulators who are keeping their internal environment as constant as possible, the ideal value for a particular parameter, like chloride concentration, we call a set point. As I mentioned previously, really the set point represents an average of values over time. And those values are going to increase and decrease a little bit as external situation changes, but the body opposes those changes to stay as close to the set point as it can. So mechanisms of homeostasis include both negative and positive feedback. Negative feedback is the most common in physiology. So this is primarily used for staying the same day to day according to some parameter. So blood salinity, body temperature, blood osmolarity. I could go on. Positive feedback is typically for adaptive responses that are designed to be, I'm using the word designed loosely here, relatively brief and intense. So you need something to happen big time for a short time. That's what positive feedback is for. So this is positive feedback under normal physiological circumstances. In medicine, a lot of diseases that you will have to contend with if you become a physician are positive feedback loops that you need to get your patient out of in order to prevent them from dying. So for example, congestive heart failure and its relationship with high blood pressure, positive feedback loop. Okay, so let's talk about some terminology with regard to feedback loops. And this is terminology that you need to understand and that I will ask you about. So the first little bit of physiological terminology is effector. An effector is an organ or tissue that basically does the changing. There are many different kinds of effectors. So the effector does something, and that affects a sensor. The sensor is then going to have some kind of a effect on the effector. So in this case, a sensor is sensing something, and let's say it's the amount of red dots, and it's detecting that amount of red dots is nominal, meaning that we have enough red dots, not too much, not too few, so the effector can hold off on producing any more red dots for the time being. We want to keep our amount of red dots steady. This is negative feedback. So another example would be, let's say the effector is in this case a skeletal muscle, and it is shivering, which is generating heat. Your temperature sensors are going to detect that heat, and once you've shivered enough to get yourself back up to 37 degrees C, so let's say this amount of dots is equivalent to 37 degrees C worth of thermal energy, then we're going to stop shivering. So either you've gone inside or you've shivered enough for the time being. Another example would be circulating levels of hormone. And I'm going to admit some bias here. Both Dr. Luderschmidt, who taught this course before me, and myself are endocrinologists, so this example works particularly well. So let's say that my effector is my thyroid gland, and this stuff is thyroid hormone. If I have too little thyroid hormone, I'm not going to use fuel efficiency. My body temperature is going to drop, and my ability to power my cells and tissues is going to struggle. If I have too much thyroid hormone, the opposite will be true. I will run hot. I will be very thin because I'll be consuming energy really, really quickly. And that ultimately causes problems as well, burning through fuel faster than necessary. So, various sensors throughout the body detect things like fuel amount, so how much stored fat you have, etc., and temp. And those sensors are going to feed back onto the thyroid gland to prevent it from secreting more thyroid hormone. Thyroid hormone itself does that also. So if there's too much of whatever the effector is doing, heating up or producing hormones or cell signalers, the sensor is going to double down on the negative feedback, making the arrow bigger. So it'll say, whoa, 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 let's pump the brakes even more on whatever the effector is doing. What if there's not enough? So the effector isn't doing its job for some reason. This is just right. That's not enough. So now we're not responding to change whatever environmental change is imposed upon us. Let's say, for example, temperature. Not shivering enough. Not burning fuel fast enough to create waste heat. Sensor is going to sense that too. And that's defined as too little. So sensors and effectors interact with each other in a push-pull kind of relationship to maintain that set point. So notice now that there's too little, the arrow got a lot skinnier. So a sensor is capable of turning down its negative feedback and that disinhibits the effector. Disinhibition is an important idea here. It's not that the sensor is actively stimulating the effector, it's just not preventing it. Those are different, and we'll flesh those ideas out more as we move along. So, assuming things are working correctly, the organism is healthy and normal, the relationship between the effector and the sensor should be them interplaying enough to keep the amount of the effector's response just right to maintain whatever parameter that is. So maybe in my first example, you always have enough heat to stay at your normal body temperature. Positive feedback, as I mentioned, is for things that are designed to be intense and brief. So first I'll explain the basic principles, and then I'll give you an example, and I think the example will help you understand positive feedback and its purpose a little bit better. So let's say the effector is producing its amount of dots and the sensor is detecting those. And instead of trying to keep the amount of dots just right, the sensor is like, I want more dots. And it gets more dots, and then it says to the effector, yeah, that's great. Still more dots. Keep the dots coming. Oops, sorry. So an example of this is the events leading up to childbirth. So in this case, the sensor would be two things. pressure receptors on the cervix. And those are detected, and then that tells the hypothalamus to release oxytocin. Oxytocin has a profound effect on smooth muscle, which the uterus is made out of. And what oxytocin does to smooth muscle is makes it contract. So, when the sensor in the cervix detects the pressure of the fetal head on those receptors, it signals to the brain to produce a hormone to make the uterus contract even harder. And then those sensors are triggered even more because the fetal head is pushing on them more, and that produces more oxytocin. And this positive feedback loop continues, but obviously it can't continue forever. So this is contingent upon availability of oxytocin. Fortunately, it's a pretty small peptide hormone, so it's not particularly difficult to make, and pressure on the inside of the cervix. So this process stops once both the fetus and the placenta have been expelled, assuming we're talking about a placental mammal. Okay, so let's discuss physiology and time. So a lot of the graphs I've been showing you, for example, are something over time. So let's discuss time on terms of its relationship to physiology, and we have a bunch of different frames or scales that we need to think about. And you can actually focus your questions and interest in physiology along these lines. So if you're interested in, say, how a salmon migrating upstream tailors its blood concentration of chloride to its environment, that's going to be an acute change, because as it swims, it's going to be experiencing incremental change in saltiness. So maybe in the short term it pumps something harder or it forms urine faster. So these are acute changes, and acute changes are reversible, meaning that if the environment stops changing, you can undo your change. Chronic changes, chronic meaning long-term, this occurs after an animal has been in a new environment for days, weeks, or months. So, for example, as it gets gradually colder, perhaps an animal gradually has its fur change from brown to white. That would be acclimatization or acclimation. You can also call this phenotypic plasticity. So phenotype is the physical manifestation of genes. And plasticity means capable of changing. Another example, let's see, I'm trying to think of some phenotypic plasticity that is especially memorable. Okay. So humans are unfamiliar with this, but for many animals that breed seasonally, they have a phenomenon called testicular recrudescence. And what this is when the animal is not in breeding state, the testicles are up inside the body and not visible from the outside. When it's mating time, however, the testes descend into and significantly enlarge in the scrotum, producing a very noticeable change in scrotum size. So one example of this change is, that's locally noticeable is in squirrels. If you pay attention to squirrels, at certain times of year, you'll notice that male squirrels have impressively large testicles, and then the rest of the year they don't. So that is an example of phenotypic plasticity as it relates to seasonal reproduction. I'm using squirrels as an example because I have a pet one and therefore an implicit bias towards squirrels, but lots of animals do the testicular recrudescence thing. Evolutionary changes are changes that occur by alteration of gene frequencies, and these are not within an individual, but rather across generations in populations. So the time scale that we talk about is larger, and within one individual lifetime, the changes are not reversible. So let's look at heat acclimation. So this is taken from a study that just looked at the ability of people to walk and relating that to how many days they had endured being in a high temperature and 20% relative humidity. So high temp, relatively dry-ish, and the endurance is being measured as can you walk for 100 minutes? Are you physiologically capable of it? And it turns out, of the group being measured, and these were particularly males, and your textbook uses a little bit of antiquated language around gender and sex here, so just keep that in mind. Sometimes I'm going to say what the graph says, and it doesn't necessarily reflect current understandings of gender and sex physiology, so just be aware of that. So, can you walk for 100 minutes successfully? And it turns out individuals that were exposed to heat longer had an easier time once they'd sort of gotten used to it. And that makes sense, right? Like if you've ever been on vacation to a very hot and humid environment, you find that when you first get there, you're a sweaty mess. And you're like, why would I even bother taking a shower? I'm just going to be sweaty again in about 5 minutes. But the longer time you spend wherever you are, the more acclimatized you are to those conditions, and you find yourself sweating less and getting less exhausted, even though it's hot. So, acute response would be things like sweating, evaporative cooling, panting, etc. But with each iteration of the acute response by attempting to do this walk, you end up with a chronic response, which is after a few days, your body says, oh, okay, well, this new parameter isn't going away anytime soon, so the body undertakes specific physiological changes to deal with and tolerate that new condition. It's also important to note that acclimatization or acclimation is not the same thing as adaptation. So this is an individual adjusting to new stimulus. An adaptation is a long-term change related to evolution, related to gene frequencies, and one or more traits. So, for example, the trunk of an elephant, this is an adaptation for the lifestyle and feeding behavior of elephants. You can look at the nose lengths of related animals, such as, for example, hyraxes, and kind of look at gradual sort of interim forms of that. So, another way to say this is an adaptation is a trait that is present at high frequency in a population. Most of the time, if it's an adaptation, by high frequency, we really mean nearly every individual has it, because it confers a greater probability of survival and reproduction. Not all traits are adaptations, and that's because not all traits experience selection pressure. You know, there are some traits that are neither good or bad, they just kind of are. And so their prevalence in a population is instead related to mutation and genetic drift. So the random frequency changes that alleles of a gene experience just because of stochasticity and because of genetic recombination. So for example, my eyes happen to be brown. I wouldn't be better able to survive or reproduce if they were blue. And I still see out of them the same. So not really an adaptation. There's also physiological variation in individual traits. So the ability to do a thing doesn't mean that all animals are the same amount of good at doing that thing. So for example, do I have skeletal muscles? Yes. Am I able to run? Sure. Can I do it as well and as efficiently as Usain Bolt? Hell no. So the adaptation of using my muscles to run exists in me and exists in Usain Bolt, but Usain Bolt arguably does that task way better. So the amount of natural variation in a trait has to be considered before you can make comparisons across populations and species, meaning that you need to get a baseline. You need to establish in that population what's the low end for a trait and what's the high end. So one example would be maximum rate of oxygen consumption. This is a figure from a study. Another example would be something I mentioned, my ability to run, for example. So who's the slowest runner as a human? Who's the fastest? So you have to establish your endpoints and look at frequencies. And if you look at this mouse, what you're going to see is that for most traits, there is a roughly Gaussian distribution where there's some value set that most individuals fall into trait-wise. And then as you move away from that average, there are outliers. Some animals that are really good at something or really bad, and I'm making a value judgment, right? So those are our outliers. Okay, so this picture has been showing you how deer mice, specifically, in a single population, deal with oxygen consumption, so how they're using oxygen metabolically under a set physiological challenge, which is what's their top speed of oxygen consumption. You can also do the same test in people, and also a little more easily, because you can take a human and say, like, hey, would you put on this weird thing and go on this bicycle? I'll pay you. And then the humans will say yes. Mice are a little bit harder to convince to work out. So in this case, we have this nice feature where some countries, as part of their public health policy, have mandatory medical examinations to help, among other things, the government understand the physical state of the population and therefore how much they can expect to spend on things like healthcare, right? That provides an awesome sample size. So 33,000, a sample size of 33,000 for a human study is really good. It's really difficult to get that big of a sample size for human studies because of ethical constraints on human experimentation. For obvious reasons, and good reasons too. I don't want to live in a world where you can do whatever to humans to find out medical facts. You know who did that? Nazi scientists. So we don't want that. But regardless, this is an impressive number. So these are 18-year-olds, obviously, for the most part, young, fit men. And they reported for compulsory medical exams. This was back in 1983, so these data are old. Also, just quick note about pet peeves. These data is correct because data is plural. You don't say this data because that is a difference in plural versus singular. So if you want to sound smart when you write, you don't need Grammarly, you just need me telling you what grammatical conventions irritate me, I guess. But regardless, if you look at the mice and compare them with the men, you'll see that although the values may differ, so let's go back a slide and compare. So we've got milliliters of oxygen per kilogram per minute, and the median is somewhere between 41 and 42. And then, we have a different metric here, so they're 19 to 20, also a very much tinier organism. But same deal, or same pattern, probably because we are aerobic vertebrates, both mice and men, albeit different sizes, and we work more or less the same with regard to lungs and blood. So unsurprisingly, the distribution is once again Gaussian, meaning bell curve shaped, tall, middle, short sides. So there are some patterns of variation that are constant or consistent across species, where others are not. It really just depends on the particular trait. Okay, so that's where we'll stop for today. The next video, we will discuss cell membranes, what's in them, how they work, and then finish up by discussing enzyme fundamentals. So what is an enzyme? What does it do? And why are they important? So thank you for your attention, and I will see you in the next video.